All right, so thank you everyone for joining today. For those of you who don't know me yet, I'm Alex, the Director of our Molecular and Genomics Informatics Core. We also have online today Hong Li from the Center of Advanced Proteomics Research, who, as you may or may not know, has a full suite of different proteomics instruments for any kind of protein structure, analysis, kind of anything you need to do, and he's a great resource if you have any proteomics questions. But for today, we do have SomaLogic here to talk about their technology, which is going to be a fun little talk. But before I turn it over to them, just a couple quick housekeeping items. The usual things. If you have any questions, please drop them into the chat, the Q&A box. We'll answer what we can live, and then anything that's not answered as we go will be answered at the end, and we'll have time for questions at the end as well. But with that, I'd turn it over to Tim and the SomaLogic team to hear about their awesome technology.
Awesome. Thanks for having us, Alex. Thank you all for joining me either today or after this is recorded. So my name is Tim Carroll. I'm the Senior Account Manager with SomaLogic in New Jersey. Today I'm joined by a couple of my colleagues as well, Will in bioinformatics and Kathy. Would you like to introduce yourself just so everyone has an idea on your role here at SomaLogic?
Sure. My name is Kathy Janko. I am part of the Global Scientific and Data Engagement team. I've been with the company for about 8 years now, originally started in the Research and Development group, and now work for the Scientific Engagement group. And so what I'm here to do is to really aid our customers in educating them about our SomaScan technology and enabling our customers to take full advantage of proteomics.
Awesome. Thank you, Kathy. And I think Will is having some technical issues, but he'll be joining shortly.
I'm here now.
Oh, you're here. Awesome.
Yes.
Will, do you want to introduce yourself and what you do here at SomaLogic?
Yeah, I'm Will Schwarzman. I'm also a member of the Global Scientific Engagement team as a bioinformatics analyst. So I'm here to talk about SomaLogic's data, some of the resources that we offer for analysis, and what support we can give.
Awesome. Thank you so much, Will. Yeah, so they'll speak a little bit later, but jumping into our agenda for the seminar, it's as follows. First, I'm going to talk about SomaLogic as a company and our core offerings. Next, I'm going to discuss what makes our assay special. And then we'll touch on how to interpret the data and the resources we provide. And finally, we'll wrap up with a mouse case study before leaving about 15 minutes for Q&A. So we're ready to get started? Awesome. All right, moving on. So who is SomaLogic? SomaLogic is transforming research in healthcare with industry-leading proteomics. There are a few key elements to establish market leadership, including menu size, specificity, and reproducibility, to name a few. I'll touch on each of these points throughout the presentation. The reason why we focus on proteins is because they provide a real-time snapshot as to what is going on in the body. The proteome changes over time where the genome is static. Subsequently, discovery efforts have shifted into monitoring circulating proteins. About 95% of all FDA-approved drugs currently target proteins. The information gathered has helped accelerate the clinical trial process by helping with subject selection, detection of subtle effects, and measuring reduced risk early. We're also gaining better insight by detecting off-target effects and ending problematic trials early. But the question is, how does this really all relate to your research? Proteomic insights are relevant at every stage of the clinical trial process. With that being said, we provide a platform that can help you achieve your goals at each stage. In the preclinical stages, our platform works with translational models such as mouse and non-human primates to help improve understanding of disease biology. Our assay has the largest menu on the market, which enables more opportunities when it comes to discovery of drug targets and uncovering the mechanism of action. With SomaLogic, you can transition from preclinical onward all on one platform. And with better coverage of the proteome, we can obtain better insights and therefore conduct more successful trials. Starting off with our flagship, we have the SomaScan Discovery. It's a simple fee-for-service model where we provide you with highly reproducible measurements in each sample. This is your data and you own it. We know how crucial discovery is in research, and SomaLogic is the unquestioned leader. The SomaScan Assay is the first and only platform that can measure 7,000 proteins in our core sample types of human EDTA plasma and serum. To overcome issues of sensitivity by other methods, we employ 3 tailored dilution bins within our assay, which allows for 10 logs of dynamic range across both highly abundant proteins like albumin to lower abundant interleukins. When you're looking at menu size, SomaLogic outpaces all the competition. And remember, with better coverage of the proteome, you get unmatched insight when it comes to biomarker discovery and drug target identification. Now that you've seen where we are, I wanted to show you where we came from and where we're going. The technology behind the SomaScan assay is based on 20 years of research. While competitors utilize some variation of antibody-based sandwich approaches, the SomaScan assay uses modified aptamers, which I'll discuss in further detail later. The unique binding properties of what we refer to as Soma reagents have allowed for the high specificity required to keep our 5% CV both intra and interplate, as we have scaled from 1,300 target proteins to 7,000 today and 10,000 by the end of 2023. One thing to note, we do have a 40 sample minimum per matrix type. We ask for this for a number of reasons. First, we want to properly power your studies. And secondly, it also has allowed us to lower the cost for our customers as we've scaled our assay across multiple generations of growth. The SomaScan platform has been instrumental in growing proteomics. In turn, partnerships with some of the top pharmaceutical companies, the NIH, and top academic institutions have followed. So you can rest assured that the SomaScan assay has been heavily vetted by collaborators and publications. Additionally, with SomaLogic, you have the option to run one of our panels. We have our custom panels, which you have the ability to select from up to 1,500 analytes from our menu. The best part about our custom panels is they're fully automated setup and they're ready to run. So you don't have to wait because you wanted to choose custom. Just select from up to 1,500 analytes from our menu. There's no added assay development costs and fees. And because we run the full 7K assay with each custom panel, you can buy up that data in the future without further validation. In addition, we offer fixed disease-specific panels in top areas of interest, including inflammation and immune, oncology, and neuroscience. Our fixed panels cover on average 9 times more targets than our competitors. Something to note, though, with the fixed panels is they don't offer the same flexibility to transition to the full 7K plex. Given the power of the platform, we later designed the SomaSignal Tests. These are algorithmically derived from our SomaScan data, enabling multiple clinical assessments from a single blood draw. This is the only platform where you can translate proteomic data into multiple clinical metrics. The 20 SomaSignal tests help researchers mitigate risk, reduce time and cost, and improve productivity. Some of the tests include glucose tolerance, VO2 max, and cardiovascular risk through a single 55 microliter sample of human EDTA plasma. This is replacing standardized methods that can be inconvenient, unreliable, or non-existent. With serum samples, the SomaSignal test can be used as a replacement to check for NASH without the need for numerous invasive and costly tests. For those that are interested in a little bit more detail on how the SomaSignal tests were developed, I included the publication from the 2019 Nature Journal of Medicine. In this study, 17,000 patients were assessed using our previous generation SomaScan 5K platform. The approximately 85 million protein measurements helped develop this 11 SomaSignal tests at the time. This proof of concept study demonstrated that using a large-scale proteomics platform coupled with machine learning can be used to develop clinically relevant health measurements. The models developed using the SomaScan platform performed as well or better than traditional models. Something to keep in mind is that these are for research use only and are currently available in only human EDTA plasma and serum. So, in summarizing the core offerings with SomaScan, you can do it all with one platform. With the SomaScan Discovery and the SomaSignal Tests, you can gain both protein data and clinical data from one sample. So jumping into our Soma reagents themselves, the secret sauce to proteomics at scale lies within our reagents. Soma reagents are synthesized from single-stranded DNA-modified aptamers. And because they're single-stranded, this gives us the ability for a universal competitor in our assay, further enhancing the specificity. They're chosen for specific shape recognition from an incredibly large and diverse library. Each Soma reagent is selected to bind to its conformational target and dissociate slowly, hence the term SOMA, which stands for slow off-rate modified aptamers. So again, here we're taking advantage of the kinetic nature of the SOMAmers, further enhancing specificity. The distinctive modifications of the SOMA reagents enable high affinity and exceptional binding through hydrophobic interactions. The high specificity has supported SomaLogic to scale its assay to 7,000 proteins and counting. Here we have a close-up look of an example SOMA reagent shown here in purple. To perform our assay, our SOMA reagents are bound to streptavidin beads via biotin, a photocleavable linker, and a fluorophore. Moving on to our assay, we first expose our SOMA reagents to our samples. The proteins are represented in the tan color. Over time, bind to the SOMA reagents, forming complexes. This is referred to as the first catch. Unbound proteins are then washed away, and we tag the protein-bound complexes with biotin. A UV light is then used to break the photocleavable linker, releasing the complexes back into the solution. For enhanced specificity, a universal competitor, as I had mentioned, shown here in green, is introduced to prevent any rebinding of non-specific complexes. Next, the biotinylated protein complexes are recaptured on new streptavidin beads for our second catch. Finally, we denature the proteins using chaotropic salt, which releases the SOMA reagents. They're then hybridized to complementary probes on an Agilent microarray slide, and the relative fluorescence is measured. So now I'm going to pass it off to Will here, and he's going to talk a little bit about the data that we provide and a little bit more about interpretation.
Thanks, Tim. So once your samples have been delivered to SomaLogic and then run, all of the proteins have been assayed and measured, then you're given a data deliverable back. And your data deliverable comes with 3 files for the SomaScan Discovery platform. You'll get a quality statement file, and that's a PDF file. The quality statement file is going to summarize our data standardization and QC process and show how your own samples and our SomaLogic controls performed on your particular run to ensure rigor and reproducibility. You'll get a scope of which samples fell inside and outside of our acceptance criteria, and then a sense of what the overall quality of your run is, everything from plate, passing, or flagging to CVs for their particular run. You're also given 2 data files. These are tabbed limited text files in our .adat structure here. So anything that can work with tabbed limited text will be able to work with these files. We give you 2 files at different levels of normalization, so you're able to choose to use the SomaLogic normalization path, use our standard reference, or if you would like to apply your own normalization to your data set, you have the flexibility to do that as well. Your data file is going to contain lots of information about your samples itself, about their same quality control metrics that are in the quality statement file, information about the plates, the plate position, things like that. Protein annotation information, so you have lots of data to subset your proteins based on, and then a matrix of RFU values that you can do all of your calculations on. Next slide. Once you have these files, as I mentioned, the data files are text files, so you're able to read them and process them in your standard pipelines. And then we have our own tools for analysis and tools to help make them work with your existing bioinformatics pipelines. We have R and Python programming packages available, which serve as readers. They recognize the ADAT data structure of our SomaLogic data and are able to read it in and create objects in R and Python that are interoperable with other packages. So if you're using Pandas or SciPy or machine learning packages in Python, or if you're using Lima, Edgear, something like that in R, our data will work with all those through the use of our packages, or you can write your own scripts to be able to process the data. Again, everything will be processed out of the text files. These packages just make it way easier to work with. We also have some example data on that GitHub as well, and these repositories are public. So if you want to go get a feel for what our data is like or start to learn using these packages, you can go to github.com/SomaLogic and begin playing around with those. We also have a web-based tool as well. If you don't have any experience programming in R or Python, or you want a more streamlined way to perform your analysis, our web-based tool allows you to upload your data into a private instance, be able to merge in your clinical metadata, filter down your data, run up to 7 statistical tests on it, and make several different types of plots to be able to visualize and analyze your data. These plots and the data generated from the statistical tests are also able to be exported out of the tool as well. So then you can take it into other tools for further follow-on analysis, such as pathway analysis.
Awesome. Thank you so much, Will. All right. So our assay provides the reliability and consistency and reproducibility for you to be confident in your discoveries. And this is achieved through our 4 keys to specificity. I know I've already touched a little bit on the universal competitor and the importance of that, and a little bit on the kinetic nature of the Soma reagents, which really enhances the specificity. But wanted to touch on the first 2 more specifically. So this is very helpful when it comes to the specificity of our assay. Number 1, because of the synthetic consistency, The SOMAs are known sequences produced using traditional DNA synthesis, and because they're synthesized, we have excellent consistency. And number 2, the controlled selection. So Soma reagents are carefully selected for optimal binding specificity from an immense library under controlled conditions. So buffer, temperature, everything is controlled, and all the SOMAs work under the same conditions. So these are some of the keys that really can help give you that confidence in using the platform, and has really allowed our platform to continue to scale while keeping that high specificity. I know we've also touched on a number of these items throughout the presentation, but I wanted to highlight a few we haven't really discussed yet. So really across the board, when it comes to the SomaScan assay, you're really checking a lot of different boxes. But what I wanted to focus on first is the speed. Speed is extremely important when it comes to research, and turnaround times are obviously very important in that. So just to kind of give you a little bit of insight into turnaround time as well, 4 to 6 week turnaround time is what we're looking for our core sample types. As I mentioned, the human EDTA plasma and serum, upon us receiving those samples in-house in Boulder. So 4 to 6 weeks from when that arrives in our lab to getting data delivery. And this is obviously incredibly important. We're looking at about 8 to 12 weeks when it comes to non-core sample types. That can vary, but just wanted to give a heads up when it comes to that. Now, when it comes to price, you're getting really incredible bang for buck and value, and SomaScan stands alone when it comes to price. At around 10 cents per analyte when running our 7K assay, you can affordably unlock more of the proteome. We have a continually expanding platform as well with best-in-class reproducibility compared to the alternatives. And all of this can be done using a single sample that can give you both proteomic insight and clinical insight. So brief overview of what we've covered today. With SomaScan, you're getting market-leading specificity across 7,000 proteins today and 10,000 by 2023. A median 5% CV across 10 logs of dynamic range demonstrates both the high reproducibility and sensitivity required in discovery. The proof is in the pudding with over 1 billion protein measurements to date and 400 publications. Here at SomaLogic, the proteomic insights gained from the SomaScan Discovery and our panels provide you with the tools you need to help drive you forward through each stage of the clinical trial process. In addition, our 20 SomaSignal tests and more than 100 SomaSignal tests in development will continue to offer additional insight that other platforms just do not have. So why partner with us? As I mentioned a little bit earlier, our simple fee-for-service model makes it quick and easy to get started with no added assay development costs or hidden fees. You own your data, but you get to take advantage of the single platform that offers both proteomic and clinical insight. And we run a high throughput CLIA lab to really help you meet your needs. And it really does take a team here. And I know we have a number of folks here. We're here to help you with every step of the way when it comes to providing consultation with study design and statistical analysis. And we're doing this because your success is our success. So I'd love to pass it off to Kathy just to kind of jump into a mouse case study. And I know we have one question coming up here in Q&A, but we'll address that after Kathy touches on a little bit of a mouse study.
Great, thanks, Tim.
Thank you.
First, I'm actually going to touch upon other sample types and alternative species. So we realize that Rutgers is a large organization and there are a variety of research projects underway. So we want to make sure that you realize the full capabilities as to how you can utilize the SomaScan technology. Our reagents were developed from natively folded human proteins, and our assay conditions are optimized in human EDTA plasma and human serum. Our assay is validated in these matrices as well as in human urine. Other sample types can indeed be run, despite not going through the rigors of validation for every possible sample type out there. A lot of value can still be realized from proteomic exploration in unique matrices. Examples are identification of biomarkers of stroke in rodent CSF, comparing the proteomic profiles in tumor tissue samples, understanding drug toxicity in cell lysates, or understanding molecular mechanisms in non-human samples. Again, the reagents were developed from human protein targets, but if that epitope is conserved, then it is possible that the aptamer can bind to its non-human ortholog. And with SOMAmer agents to nearly 7,000 unique human proteins, one can still cast a wide net for exploration. Next slide. And so here is a list of some of the samples that customers are running on our assay. You can see human EDTA plasma and human serum and human urine as our core sample types and the volume requirements that we ask from the customers. For non-core sample types, customers are running things like non-human primates, mouse samples, cell lysates, tissue homogenates, aqueous humor, vitreous humor, CSF. The list goes on and on. If you have a sample type that you do not see on this list, please feel free to reach out to us. We are always happy to help discuss the intricacies of unique matrices. Next slide. Thanks. And so now I'm going to go into a specific case study involving mouse samples, where the objective is to understand biochemical pathways of muscle and bone loss. Next slide. So just to give a little bit of background information, myostatin and activin A are signaling proteins involved in regulating the growth of muscle mass. These proteins maintain muscle homeostasis by limiting muscle growth. Animals that have been shown to not have the myostatin gene exhibit dramatic muscle growth, up to 2 times more muscle density as wild-type normal animals. Muscle and bone mass are known to be significantly diminished during prolonged spaceflight, which is a problem for astronauts. Several attempts have been made by pharmaceutical and biotech companies to develop myostatin inhibitors, and these have been tested in human trials, but none have really come to fruition in approval of a drug for actual clinical use. So in this study, a different approach was taken by introducing a decoy receptor known as ACVR2B Fc. This decoy receptor traps myostatin and activin A, making them unavailable to bind to their true receptors and effectively cutting off the pathway for muscle inhibition. And so the goal of the study was to understand the various pathways affected when targeting myostatin and activin A with this decoy receptor in mice on Earth and in mice after spaceflight. So as for the experimental design itself, this involved 2 cohorts. The first being a group of mice that were sent into space for about 1 month, and another group of mice that were earthbound. Within each of these groups, you had the normal wild-type mice, you had mice that were injected with this decoy receptor, and you had mice that lacked the myostatin gene. Mouse plasma samples were run on the SomaScan Assay 5000-plex. RNA-seq data was also generated from bone and muscle from these mice. And some ELISA experiments were also tested with plasma samples from these mice. So findings. What we're clearly, these normal mice, these wild-type mice, they suffered significant muscle and bone density loss in space. Those that were treated with this decoy receptor showed that showed significant muscle growth and maintained and grew their bone density. This was in both earthbound mice and in mice exposed to microgravity. Ultimately, what was found was that there were numerous proteins and signaling pathways that were related to muscle and bone homeostasis. Next slide. And so here are some of the proteins and the actual pathways that were altered due to exposure in microgravity and treatment with the decoy receptor. The plots on the left show the log2 ratio of SomaScan protein levels in grounded treated mice to untreated mice, or flight to ground untreated mice. The proteins affected were involved in pathways that had to do with bone resorption, muscle growth, bone and muscle homeostasis, and inflammation. And the various responses, the upregulation or the downregulations, all made sense from a biological standpoint. It's also notable to mention that these findings were also supported with other platforms. TRAIL and RANK-L, which were significantly upregulated in plasma proteomic data, are also upregulated in response to treatment in RNA-seq muscle and bone data, as seen in the bar graph labeled B in the top right of the slide. Also worthy of note is that the signaling pathways identified in the SomaScan plasma data were also found to be implicated in RNA-seq bone data. Myostatin concentrations were also measured by ELISA in ground and in space flight treated and untreated mice. This is shown in the bottom right corner of the slide under C. Myostatin was downregulated with treatment, as you can see in this ELISA, which makes sense because the proteins were being cleared by binding to this decoy receptor. This is consistent with the SomaScan data, where myostatin was also downregulated in plasma of treated mice. And so here you have this multi-omics and multiple platform approach that all support each other. Next slide. So how was SomaLogic technology leveraged? By simultaneously studying nearly 5,000 unique proteins in the SomaScan assay, this enabled researchers to understand multiple signaling pathways involved in muscle and bone regulation in microgravity. And as a result of blocking myostatin and activin A pathways, the data can possibly lead to ways to protect against muscle and bone loss during spaceflight, in addition to utilizing a therapeutic strategy to help people on Earth who experience muscle and bone loss. For instance, people in wheelchairs, people who are bedridden, people who have muscle atrophy or are or have muscular dystrophy. Even though the SomaScan assay was designed for humans, there was meaningful and impactful information gained in this non-human study, which was supported by other experimental approaches like RNA expression analysis and ELISAs, which really emphasizes the power of the SomaScan assay technology. So numerous studies have been performed on the assay in a multitude of sample types, as well as in non-human species. This example specifically focused on understanding proteomic pathways and mechanisms of action, but other applications involve biomarker discovery, preclinical research, effects of toxicity for drug treatment, drug development. All of these things are possible in these other sample types that would otherwise not be possible in, say, human plasma or serum. So, as you can see, SomaLogic technology can be used for a wide array of applications and a wide array of matrices. We would love to learn more about your research and discuss how SomaLogic can take you to the next level of exploration.
Thank you so much, Kathy.
Thank you.
Yeah. I believe we have one chat here in the Q&A from Hong. How much does it cost for 40 samples at 7,000 proteins? So I believe the list price for academics is, I believe it's slightly less than for pharmaceutical. I know for pharmaceutical it's $750. I believe it's $700 for academics. But I'd have to take a look in that. Give me one sec. I think it is that though.
That's per sample, or is that the total price?
Per sample. Yeah, it's per sample. Yeah, it's $700.
I was gonna say that's the entirety of it.
Yeah, so you get the entirety of the menu. It would be $700 per sample, and you would unlock all 7,000 analytes in human EDTA plasma and serum.
You say 700, I have to times that by 40, correct?
Yes, I believe that would be 28,000.
Okay.
If my math is correct
And,
there.
and the 40 is the minimum number of samples per project you guys will accept, if I hear you correctly.
Correct. Yeah, that's what we ask for. We try to, have that just to properly power the study. Occasionally, we've seen some that have gone slightly below that. But we also want to set you guys up for success. We don't want you to have, a study run with, 12 subjects, and then the data comes back and it doesn't offer the same sort of impact for your studies, because then, you would say, what's going on here? Like, maybe we should have had more samples, and we know how valuable your samples are. So we really want to just maximize, the work that you guys have. So that's typically why we ask for 40 as the minimum.
Yeah. Okay. And the next question is, biologists, when they do discovery proteomics, they like to validate their results. And because your data is discovered with aptamers, so how reproducible are your findings can be validated by antibody or imaging techniques that biologists are familiar with?
Right. So absolutely, we encourage validation of the SomaScan data results to orthogonal methods, ELISA, Western blots, so on and so forth. That can give you additional confidence in the outcomes. However, it's important to understand that SOMA reagents and antibodies are different. They can measure slightly different things. So our SOMA reagents are measuring the availability of an epitope on a protein. If there is a change in the epitope for whatever reason, that is going to be reflected in the signal. And so there will be instances where this kind of sensitivity is not picked up in other orthogonal validation methods. And these are things that you have to take into consideration when you're seeing a lack of correlation between the two.
Also, I would like to add one point. By the way, guys, my name is Tala. I am the Director of Scientific Engagement here at SomaLogic. I crashed this meeting to learned. I just like to add one point that Kathy perfectly explained. First of all, our assays go through the various validation methods. Many of them go through mass spec for the validation. All of them go through the visual validation running the gel. There are so many levels of the validation that each of the assays go through. But if you think about it, our SOMAmers are so specific to the form of the protein that we make sure that if there is any PTMs or any modification of the protein that happens, we catch that. In other methods that if, for example, you use the other orthogonal validations like ELISA, not all the antibodies as was presented in the team's presentation, not all the antibodies have the same level of sensitivity to the protein modifications. For example, polyclonal antibodies, by the name of them, they are insensitive to a lot of the protein conformation or other methods that you are getting it. So it is important that when we are talking about orthogonal validation, we understand that what validation already they have gone through, that they are actually very extensive right now that you can see from our validation list. But if the method that you are using, does it have the same level of sensitivity or not? Or is it going to miss certain information that the SOMA is going to provide for you?
Yeah, those are very valid answers. We, as a biologist, we have no way to question the validity of your assay because it is new. The problem is when you publish, so I run a proteomics, a mass spec-based proteomics core. So whenever we do TMT proteomics, mass spec proteomics, people always want to validate their results, typically with antibodies. So all the explanations you offered is totally valid, but that still doesn't address biologists. They are familiar with antibodies. They are familiar with imaging techniques. So unless, I guess, SomaLogic provide aptamer as a analytical reagent for imaging or for validation. Let's say, I don't know whether you can use aptamer for Western blot, for example. So unless you provide follow-up reagent or alternative aptamers that differ from your discovery aptamers for validation, people will question the results. I mean, for good reasons, because when people compare SomaLogic results to another high-level platform Olink, the overlap, for the most part, is not as big as people want to believe because they use antibody-based techniques. So you can argue which is better, but the reality is biologists has to validate results from high throughput analysis. So the purpose of my question is, what kind of aptamer-based reagent you will provide for people after they discover interesting changes from the high throughput analysis?
And I will allow the team to answer that question. I just want to add one point here. We always welcome the validation, regardless of the method. If you're using Olink or you're using Soma, it's never a good idea that you limit yourself to one method of looking at it. You have to always confirm your finding. Again, for many of our assays, we have done mass spec. We think that that is a superior method. Our data correlate very well with the mass spec. There are publications out there, and we can provide with you with the sequence of the target amino acid that the epitope and the region that SOMAmer binds to the protein. So you can, when you are selecting, for example, ELISA, for example, which we don't think that is a perfect method to choose because of what we discussed. If you are choosing, you make sure that your antibody is targeting the correct epitope, that it's targeting correct, because as we all know, proteins, the regions of the protein can have different functionality. So we provide those information for you regarding the target sequence of the amino acid. So you can select the orthogonal validation, whatever method that you are doing, you do it correctly. Apart from that, I will allow the rest of the team to see that what additional material we provide for the validation.
Yes, so we do have a SomaMer reagent program where you can order SomaMer reagents for follow-up validation experiments. And we have some suggestions, some methods that we can provide, some guidance that we can provide to you. Mass spec is kind of the gold standard, as you say. However, there are some limitations to even that. Proteins that are in very low abundance, you're gonna have a problem with that. But if there are other ways, other methods to do this validation, then yes, we are able to provide those reagents to you.
Yeah, and as Kathy was saying also, specifically in regards to mass spec, we validated 5% of our menu with mass spec, but because of some of the pitfalls, as she had alluded to, we could only validate so much of the menu. So, we feel like, the 10 logs of dynamic range without depleting some of these proteins is a strength of our assay. But we've continued to validate through other methods, through validating the biosimilars from the FDA and so forth. So, we'd welcome any sort of validation, and we definitely have some more information we could provide that I think would maybe put you a little bit more at ease.
Thank you.
Awesome. Do you have any more questions for, I guess, our panelists at this time?
Doesn't look like we have any right now, but we can provide your contact information. So if anybody has questions down the road, they can reach out to you, right, Tim? To you and the team.
Yeah, I can definitely send Alex the slides. That way, folks that joined or watched this subsequently, will have access to that. That'll also have like my contact information as well. If any of the other panelists are better suited to handle the question, whether it's our technical support team or Will in bioinformatics, I can also help direct that, that to the appropriate party. But we really appreciate everybody hopping on and listening in.
Yeah, thanks for giving a great presentation. It's always great to hear a new technology that we can see as an option. Scott.
May I ask two last questions
Sure.
since the attendee is not asking? One is I saw in January you guys are building some relationship with Illumina. So will that next-gen sequencing technology change your microarray platform? So is there anything that you can disclose that allows us to see the future of this technology? Because
yes.
that Illumina collaboration is public information, correct?
Correct. Yeah. So I'll give you my answer on it, and if anyone wants to jump in and add anything else, like as far as what you know, that's what we know at this point. So we really talk about our current assay. We try to give you a little bit of a look behind the curtain and kind of speaking to, hey, like in 2023 we should have 10,000 in our assay. That's our plan at this point. As far as the relationship with Illumina and how that's going to change how we operate at SomaLogic, I don't have anything else to add for you, but I think it would be an exciting thing for us and continued strength for the platform.
Great, because many labs, many schools has Illumina machines. So if let's say if your reagent is compatible with Illumina next-gen sequencer, maybe we can do the analysis perhaps cheaper locally rather than send sample to you. Would that be a possibility?
Yes. So I do know that we do have some facilities, and hopefully I'm not speaking too much of for myself here, but we do have some universities and companies that do have like kit sites that do a ton of samples, and they're able to kind of get set up where they don't have to send samples to us here in Boulder. That can kind of be set up, locally on site. I know Rutgers having multiple campuses all across New Jersey, that could potentially be more helpful than instead of having to ship samples all the way across the country. So I think that, that's another conversation that we can have if that's something that Rutgers is looking into. That way, no matter the campus, folks could have easier access to the technology. But if anybody else wants to add anything to that, the floor is yours.
My final question is your aptamer selection. Obviously, you guys do a positive selection for DNA binding to native proteins. Do you do negative selection first? And secondly, how do you distinguish phosphorylated version of a protein versus the non-phosphorylated version. So do you make a lot of native, how do you get those native proteins? Is that from mammalian cells or from E. coli? And the purity of those proteins will dictate how specific your aptamer will be. So if you can share, simply how that works.
Yeah. So I guess I'll answer the second part of your question because that's more recent. But our proteins that we use in the development process of SomaMarie agents in the SELECT process, these are a variety of recombinant proteins that are synthesized in mammalian expression systems, bacterial expression systems, insect expression systems. We also have a purified protein from say human serum and isolated from human biological samples as well. And so, right, so there, you made a point about how about the purity of the recombinant protein itself. And yes, so we're aware that purity can be an issue. We're constantly running pull downs to see pull down analysis and looking at these proteins on gels to understand the purity. So in pull down analyses, we can physically see the SomaMarie agent binding and pulling down the protein target band as opposed to and make sure that it's not pulling down or binding to impurities. The other things that we do in the screening process of each of the SomaMarie agents is to screen multiple SomaMarie agent candidates in human plasma titrations, spike and recovery experiments. We're looking at we're looking for the KD, the affinity of each of the SomaMarie agents. And so that really enables us to whittle down this list, find the SomaMarie agents that are performing optimally in biological matrices. So you can imagine that a SomaMarie agent candidate may be may go through this screening process, but lo and behold, it's not resulting in a great buffer titration in an actual plasma sample. And so that's a red flag for us that right, maybe the SomaMarie agent is not binding especially well to the target protein, but maybe binding to an impurity of some sort. And so that's going to be that's not going to make it through the screening process. It's not going to end up on the menu itself.
Thank you.
Mm-hmm. And then your first question was it had to do with screening for DNA or
no? My question is, how do you? Two questions. How do you distinguish phosphorylated form of a protein, PTM containing protein versus, because PTM is dynamic. Just because you purify from a mammalian cell doesn't mean a particular protein is modified. So
yeah. So we generally so the SomaMarie agents generally are not influenced by PTMs. It's possible that if a PTM really is large enough or in this conformation that is able to really affect the epitope, yes, there may be like theoretically possibilities that the SomaMarie agent would be affected by the PTM, but largely we do not think that they are sensitive to PTMs. And in terms of distinguishing PTMs, our SomaMarie, we would have to have had developed a SomaMarie agent against a protein that has a PTMs and a SomaMarie agent that does not have the PTMs. This is not something that we have done extensively at this point, and so we cannot make that distinction in the current assay.
Thank you.
Awesome.
Cool. So yeah, I guess if any more questions, Hong? No? Okay. All right. Well, I guess if we don't have any other questions, thank you everyone for joining, and thanks for the great seminar from the SomaLogic team. We'll get this posted over the next couple of days, and then if anybody has any questions, feel free to reach out to myself, to Hong, to Tim. We'll send out the contact information for everybody, and look forward to seeing everyone at the next seminar. So thanks, guys.
Thank you, everyone.
Thank you.